Hello, everyone. Welcome to the 2026 Great Lakes BioBlitz webinar. We're so glad you're here. This Great Lakes BioBlitz is science in action, and it's put on by the Center for Great Lakes Literacy, also known as Seagull. In this 40-minute webinar, learn about observing nature from a local scientist and gain some tips about using iNaturalist for data collection. Project partners will share some resources for land water learning across our Great Lakes Basin. This webinar will be recorded and links will be posted to the Minnesota Sea Grant and Seagull websites.
Educators, students, families, youth groups, and the general public are invited to participate in the 2026 Great Lakes BioBlitz. A BioBlitz is a community science effort to record as many species within a designated location and time period as possible. The BioBlitz focuses on the entire Great Lakes Basin, US, and Canada. It runs for 4 weeks, April 22nd to May 24th. Some people try to get a plant or animal each day at their school or in their neighborhood, while others go on a special hike or complete field study trip. The observations are made using the iNaturalist app, and this webinar will teach how it works.
That's right, Anna. A BioBlitz provides a way to record biodiversity, the variety of wild living things found in any given area. For the Sea Grant Great Lakes BioBlitz, residents of the states and provinces in the Great Lakes region work together to collect as many observations as possible. So you might be wondering, what is Sea Grant? And what is the Center for Great Lakes Literacy? Well, the Center for Great Lakes Literacy, Seagull, is a collaborative effort led by Sea Grant educators throughout the Great Lakes watershed. One way to think of a watershed is the land where the water flows across and down. Sea Grant is connected to your Great Lakes State's University and serves people who need water science. Check out this cartoon that shows just some of the ways that Sea Grant and Seagull provide education and research data to industry leaders, policymakers, water science to educators, community organizations, water science to municipal governments, university stakeholders, and people of all ages like you and me. If the needed water science doesn't exist, we seek to fund it and work to ensure it gets to those in need.
My very favorite thing about Lake Superior is how incredibly large it is and that the beautiful lake's water is deeply connected to so many living things. From small critters to plants and huge ones, all of the BioBlitz observations that we collect help us better understand land and water connections. Biodiversity data can contribute to research and conservation efforts.
My name is Heidi Ferris, and I'm with Minnesota Sea Grant and the Center for Great Lakes Literacy. I brought some friends with me today. Welcome Anna and Jam, the striped skunk.
Hello, my name is Anna Neal. I am the Community Learning Manager at Great Lakes Aquarium in Duluth, Minnesota. Here at Great Lakes Aquarium, we connect all people to the waters of Lake Superior and the entire world's waters, from people like you all across the Great Lakes Basin. We accomplish this by giving our guests close encounters with fully aquatic animal ambassadors, as well as land animals and semi-aquatic animals that live in and around our watershed. I brought one of our ambassador animals with me today. This is Jam the skunk. Jam and his brother Jelly arrived at the aquarium 4 years ago as kits or young skunks. They were born in human care and had their scent glands removed as babies. Jam and Jelly help visitors understand a bit more about how important skunks are in our ecosystem. Skunks are great at pest control. They eat beetles, grubs, wasps. They also help aerate the ground through their digging behaviors. Striped skunks have a large range, primarily made up of the Great Lakes states and Canada. This time of year is an especially busy time for skunks as they become more active after a long winter. Have you seen or smelled any skunks recently? He's eating some broccoli right now, getting a little bit of enrichment there. Students, learners, and scientists like you all can help animal populations by entering any sightings into iNaturalist, including skunk sightings. Biologists and researchers regularly check iNaturalist to see where and when skunks, turtles, birds, butterflies, trees, flowering plants, and so many of our Great Lakes species have been spotted. This sharing of observations through iNaturalist helps them to save time and resources when doing their conservation work.
I'm here in the St. Paul area, and you're at the Great Lakes Aquarium up in Duluth, and we have people joining us from across the Great Lakes Basin. But Anna, what does a skunk have to do with the Great Lakes?
That's a good question, Heidi. Striped skunks like Jam live in and around the waters that flow into the Great Lakes. And it reminds us that all of us are a part of the land-water connections. Water is in the Great Lakes, it's in the air around us, it's in the sky above us. Water flows around, flows across the land, sinks in it, and moves through living things and impacts us all.
So insightful. Thank you so much. And wow, is it just fun watching Jam just cruise around there snacking on things? Thanks for the reminder also about the water-land connection and the call to action for us to care for the interconnected hydrosphere, biosphere, atmosphere, geosphere. This 2026 Sea Grant Great Lakes BioBlitz on iNaturalist will happen over the next 4 weeks. And I think it's really going to help us tune into those ecological systems. For the folks tuning in to this webinar, we can all make observations right where we live. And then by adding our photos to iNaturalist, we'll be able to really tune into those systems in the Great Lakes Basin. So this webinar is going to teach you how to do that.
Yes, and if you haven't done this BioBlitz before, please know that later in the webinar, we'll share details about how to use iNaturalist, so stick around. I'm going to get this cute little skunk back into their important role here at the aquarium. So thank you, Jam, for representing your species and helping the aquarium educate so many people on the importance of water and land conservation.
And now we have an expert scientist who knows all about the importance of data collection. Please meet Angela Gupta from the University of Minnesota. Angela is an extension educator who specializes in forestry in the Rochester area. Take it away, Angela.
Great. Thank you so much, Heidi. All right. I am hopeful that you can see my slides. Yes. Okay. So I'm going to talk to you about using iNaturalist for participatory science and a lot of examples from what I do, which is University of Minnesota Extension Forestry. So first, I want to define what participatory science is, and it's science that gauges the public in advanced scientific knowledge by formulating the research questions, collecting the data, and interpreting the results. And so my projects typically include volunteers and participatory scientists in all of those features of science. And then these, I think, 4 slides are from iNaturalist directly, and they're going to introduce just a little bit about what iNaturalist is, because that's the app that we want you to use in your Great Lakes BioBlitz. So let's get to it. So there's a huge challenge out there. The climate is changing, habitat is changing, a million species face extinction, and we want to engage, iNaturalist wants to engage people to gather the information needed to guide conservation. And this is where you will absolutely be a part of the solution. So the more people we have out there looking at the natural world, in the more places we have, the more we learn. And so we can learn about all kinds of different species anywhere in the world using the free smartphone app iNaturalist. So how does that help using your phone? It engages people in nature. That's always awesome. It allows anyone with a smartphone and camera to explore nature and contribute to science and conservation. It accelerates biodiversity science and conservation. And then iNaturalist is able to crowdsource the world's largest database for biodiversity. And so I'm going to offer you several examples of how we do that in extension forestry. Use the crowdsource information from iNaturalist to inform forestry science and understand our changing ecosystem. So I do that through this participatory science projects, and I do a lot of invasive species projects and resilient landscapes. So thinking about the future climate of Minnesota's forests and how you can be a part of that. So I'm going to highlight a few that are quite specifically using iNaturalist here this spring in 2026. So golden oyster mushroom is a mushroom that is not native, and this picture here on the right is from iNaturalist of its. These are reports or observations of golden oyster mushroom in Minnesota. And it's non-native. There's some early research showing that it is likely to be invasive. It's going to cause economic, ecological, or human health harm. And so we're really trying to understand its current distribution and density across the state. So the call to action is for anyone who sees golden oyster mushroom to report it to iNaturalist. And we are most interested in reports up in northern Minnesota where we have none. You guys are concentrating on the Great Lakes, so all this area here in our Arrowhead. And then for this case, we would also like reports in southwestern Minnesota. Another one, this is an interesting case study. So there's an invasive beetle, Japanese beetle, and then there is a biocontrol fly called winsome fly. And this little dot is the winsome fly eggs on Japanese beetles. And so this participatory science project is asking people to look closely at these invasive Japanese beetles, and if they see the winsome flies, to then report it to iNaturalist. And it turns out that we've learned a lot, and we now understand from this is the third year that we're asking volunteers to do this, that these winsome flies are expanding their range across other areas of Minnesota and Wisconsin and Iowa that have Japanese beetles to help reduce the numbers of those destructive beetles in the landscape. So it's a tiny little egg that you're looking for on not a terribly large beetle, but there's plenty of evidence that folks just like yourself can help us do this by reporting, paying attention, and then submitting those observations to iNaturalist. So now I want to tell you about some really exciting work that we're doing in Extension Forestry, this climate ready woods work. And I'll spare you a lot of the back end details, but we ended up creating 4 different tree and plant lists for each of Minnesota's 11 ecoregions, so 44 lists. And one of them is a near native list. So things that aren't native in a specific ecoregion. So let's say ecoregion 2, the Arrowhead of Minnesota along Lake Superior. So some examples might be black walnut. So black walnut is native in southern Minnesota, but it's not considered native up in the Arrowhead. And so we're trying to understand this species is expected to do well in our future climate. Is it already there? How much? Is it able to survive? That kind of thing. And we can use iNaturalist to help tell us those bits of information. So we started this project in 2024. And so these first couple of slides, we are still crunching the 2025 data. So there's actually tons of data, and it takes us a while to work through. So I don't have 2025 numbers. So you're going to see a combination. So this screenshot is of an iNaturalist project set up to do this work. And you can see we had over 9,000 observations, so individual reports of these near native species in the state, representing a total of 31 different species. This is identifiers. 567 different people helped to identify, to verify those species descriptions. And then 2,700 individuals submitted observations about these species. So we are learning a ton thanks to the participatory scientists just like you. And for this project, you don't have to do anything special. If you were to find one of these species, you report it to iNaturalist, then iNaturalist on the backside will scoop it up and show it to me in this project data.
Angela, this is so fabulous to hear these specific examples of participatory science in action, the data collection at work. As you continue to share about iNat stats in action and other evidence, I just wanted to also share with those joining in on the webinar that if you have a question about the 2026 BioBlitz or iNaturalist for Angela, please enter them into the Q&A section, and we have another person monitoring. So then when Angela is finished sharing with us even more ways that she uses iNaturalist and data, then we can come back to those questions. So thank you so much, and continue on, Angela. This is great.
Yeah. No, thanks, Heidi. I'm glad you sent everyone to the Q&A because that's a great way to do it. And I do hope you guys are engaged and you'll have questions at the end. Okay, So we in this tree tracker project, the data from 2024, which we were able to crunch. So at that time, there were over 3,000 research grade support submissions, and the research grade is what happens is iNaturalist, the app, uses artificial intelligence to try to categorize or identify the species based on your photograph. So good photos are really critical. And then the second level of verification is a human in the iNaturalist community will go in there and agree or disagree with the original species identification. And once there is agreement, then that report or that observation becomes research grade. And then folks like me definitely use that information. So in the first year, 2024, we had over 3,000 research grade submissions, and those came from 318 different participatory scientists, and that represented 231 new to region observations. And so here is a map, and then the red ones were of particular interest to us. So those were new to region observations in the areas of greatest interest. So, and then this, we are also thinking about rewilding in this, and I have a slide in a few minutes that will define that. But this iNaturalist project, we were able to establish some indicators of rewilding. And so, as I mentioned before, this is Eastern Black Walnut. This is the iNaturalist page. Here, this was from 2025, so a year ago. Here were the reports. And then this is what the DNR generally considers to be the native range of Eastern Black Walnut. But these two reports came in during our project, and they're clearly much further north, and they're in this area, this ecoregion that for which Eastern Black Walnut is considered a near native. So it's of great interest to us. And then we looked a little bit closer, and this one way far north up in Superior National Forest near the Canadian border. This is the entire iNat report. It was a single individual image of an Eastern Black Walnut nut, and the nuts are quite definitive. It's easy to identify that, so no confusion. So AI said, or the reporter said, yes, it's Eastern Black Walnut, and then two different individuals said, yep, it is, which makes it research grade. But what makes this super interesting is not only is it Eastern Black Walnut, it's a mature Eastern Black Walnut. It has been somewhere nearby long enough to produce nuts. So now it's producing, it's seeding. And then you can see that nut has been nibbled on. So something locally, way up near the Canadian border where Eastern Black Walnut is not considered native, is using Eastern Black Walnut. And so that is evidence of rewilding, which I'll talk about in a moment. So we were super excited to see this all from a single image. And then I love iNaturalist because you can see someone asked, do you know whether this source of this tree was wild or could it have been planted? It's a really great question, and I'll talk about cultivate and captive in a bit. But for this project, for the Climate Ready Woods project, I don't really care. I'm interested to know, can that tree survive? Is it able to get to maturity? Are things using it? Can it be a contributing member of the ecosystem? And so this was great. It was a great report. Another example that came through this year. So again, we're still crunching the data, but there was a huge effort after the end of 2025 to run all of these through and make sure all of our different observations got to research grade. And so we had volunteers doing this for us, and we had like iNat verification parties with professionals just to get through all of those, 9,000 reports. And we ran into this one. So American sycamore is native to Iowa. It's not considered native to Minnesota. It is considered a near native in a number of different ecoregions, mostly in southern Minnesota. And so we're very curious how it's going to do here. So here is the picture. And then here is where it is. It's in Minneapolis. And then this was super interesting. So one of our volunteers said, hey, can you provide more pictures, pictures of the whole tree, the leaves, the twigs, because the University of Minnesota were really interested in the species for our Climate Ready Woods project. So I love that. It helps us do greater identification. But we also noticed this. So one of the images had note, this American sycamore was part of NASA's Moon Tree program. It was planted in May of 2024, suffered transplant shock, but bounced back beautifully this spring. And so all of a sudden we realized that one of our Climate Ready Woods near native trees, a part of our tree tracker participatory science project, was also a part of a NASA project, which just felt super cool. And so we were only able to make all of that connections because of people all across Minnesota and the world using iNaturalist. So it was really fun and exciting for us to see that. So another way in which we use iNaturalist is this is a park near my house in which I volunteer, and I have created an iNaturalist project. So anyone who goes out and iNats in this park, their observations will show up in my project. And this park is a city park. It has a beautiful manicured garden, so a lot of weddings and prom photos. But it also has some kind of more neglected woodlands on the slopes, and that's where I spend a lot of my time. The background image here, the bluebells, that's from that area, and their bluebells are in bloom right now. So it's a beautiful time in the woods. So you can see in this iNaturalist project, there are over 130 total species that have been observed on this site. And that is representing, actually went up to 140 here, 235 different individual observations from 14 observers. So not a ton of people are iNatting, but then some of my colleagues that do a lot of invasive species work has said Plummer House property probably has the highest density and most variety of invasive species in the state. So that's not exactly the kudos that we want when we're working on a site, but alas, that's the state of the state. And I have identified through iNat and a few other ways a bunch of these species. So here they are, and some of them I can try to physically get rid of, and some of them I can't. So we don't know how to get rid of jumping worms, non-native earthworms, golden oyster mushrooms, honeybees, which aren't considered invasive, but they're non-native, emerald ash borer. So what I have been doing though is removing in a lot of invasive plants. So you can see back when the pandemic started, school was shifted to online learning, and my kids' soccer all got canceled. And so my two boys and I, we went out to the woods and we started removing these invasive plants. And while I'm out there, I'm iNatting. I make it a point to at least iNat one new thing a day when I'm in the woods. And so, it just adds up over time. And then jump ahead, 5 years later, my boys are back in all their sports. One is away at college, and mostly it's just me out there. But you can see this real stark difference in the understory because we've gotten rid of so many of the invasives. And what I realized that I knew that I was enabling natives by removing invasives. That I knew, but I was really struggling to make sense of what I was experiencing in the woods. And so I had been teaching this climate ready woods work, and I'd been talking about rewilding, which is this idea of that an ecosystem can look different in the future based on our future climate instead of restoration, which is kind of taking it back to a state from before and a climate that may not be our future. And so it's looking at a new combination of species and interactions in those species, and can those still meet the ecological needs of an area? So if you go back to like third grade in your tropic triangle, and you have plants on the bottom, and then you have insects and birds and small mammals, and then you have your big carnivores, can you meet all of the needs of an ecosystem, even if the critters and the plants are a little bit different? So I was like really frustrated. I couldn't figure out what native plant community this was. And so this is that rewilding slide. Really quickly, rewilding is increasing biodiversity, restoring natural processes. Rewilding aspires to reduce human influence. Rewilding is open to novel and emerging ecosystems. So it is creating a resilient, self-sustaining ecosystem for the future. And then I had this epiphany last spring that park in which I'd been working in since 2020 was an example of a novel forest, of a rewild ecosystem. So then I decided to do a little digging, and I had pulled up before the aerial photo from 1940. So you can see Dr. Plummer was an original founding doctor of the Mayo Clinic, and this was his home. And you can see this was the original driveway, and this was a goat prairie on this south slope. So this is south facing. It was dry, sandy, hot. This is north slope, so it's cool and moist. And then the formal garden is up near the house. And if you jump forward to 2025, you can see that it's all forest. So through essentially benign neglect on the forested hill slopes, it has filled in with trees. And that's because as we get warmer and we get wetter, trees are what this ecosystem wants to become as the dominant feature on the landscape. And so, okay, there are a lot of trees. But I couldn't figure out what native plant community it was. And that's when it dawned on me this spring, it wasn't a native plant community. It was a rejiggering of the world. So I went back to my iNat posts and I pulled all of the things together and I realized I really had a unique and unusual ecosystem. So there's a stand of fairly young eastern hemlock trees. They're also threatened in the state of Minnesota, and they're considered near native. And here they were at the Plummer House. I had a bunch of Norway maple. These are considered fully invasive. I strongly suspect it was planted as a driveway tree, and then they started reproducing aggressively, so now there's a whole stand of them. A lot of white pine that's still up on the top of the slope. This is super interesting. American sycamore, that moon tree, that same species. So here's one in the woods that's full size. It produces seeds every year. It's clearly been there for a long time, so that was interesting. And then we have black walnut. Now that's projected to do very well in our future climate. It's native to Southeast Minnesota. It's a great wildlife tree. I had some of those. Then I'm out there iNatting, right? At least one iNat observation every day. And so I was paying attention to other things. So the understory, this is super interesting. There's an Ohio buckeye. It's considered near native. It's not on any of our lists. I don't really know how it got there. I suspect there's one in the neighborhood, but I've yet to find it. And that is actually reproducing. So I think the squirrels are moving around the nuts. The nuts are growing into small seedlings, and the seedlings are not getting eaten by deer. So that's actually quite interesting. There's some wild cherry, pagoda dogwood, red elderberry. These are natives. They're all projected to do well in our future climate. So it kind of makes sense that in a neglected woodlot, you're seeing things that are likely to do well. So that's all in the north slope. Then I went to and then there's some things that aren't desirable. So invasive jumping worms, can't figure out how to get rid of those. So the ecosystem is different. It's not what it was before. Then on the south slope, so this is dry and warmer and sandy slope. And I've got some eastern red cedar. That makes sense in that old goat prairie. Got more white pine, some more walnut, tons of honey locust with these gigantic thorns. That's like super curious. And then the dominant species on that slope, hackberry. And hackberry tends to be more of a mixed hardwood. It's not necessarily a dry sandy site species, and yet that is the most common tree on that slope. And that's what was really like eating at me. There's no such thing as a hackberry forest type. That's not a native plant community. But hackberry is projected to do very well in our future climate. Also some black maple, and then lots of little things, because again, I'm iNatting every day. So all kinds of interesting mushrooms, columbine, jack in the pulpit, woodland sedge, false Solomon seal, trillium. And then I realized I also had the tropic triangle information. So the critters that were using this. And so all but the bald eagle picture was taken at the Plummer House using trail cams mostly. And each of these species has been identified at this small urban park very close to downtown Rochester. So tons of deer, lots of reproduction of deer, fox eating rabbit, a fisher. This was fascinating. So when my trail cam picked up this fisher, fishers were extirpated from the state. So they were pushed out into Canada, and they've been slowly working their way back from the north to the south. And when that picture was taken, this was the southern and westernmost sighting of a fisher in the state of Minnesota. And so it was quite exciting. I iNatted it, and then I sent the iNat link to my wildlife professor colleague in Crookston, and said, oh, "Is this really a fisher?" And then very, very quickly on iNaturalist, I got contacted by the University of Minnesota Duluth researcher studying fishers, and he said, "Are you positive of your location?" And I said, "I am. I know exactly where that wall is." And so all through iNat, we were able to, within hours, confirm it and get it included in our research project. So then, of course, we have raccoons. A nesting pair of bald eagles moved into a white pine at the Plummer House last spring. Now, sadly, their nest fell down later in the summer, and so it's unclear to me if they found a new home at Plummer House. But they moved in last year, and we're hoping they come back. Tons of raptors, lots of squirrels, lots of turkeys, lots of rabbits, which are like the base of the food chain for everybody up above them, I think. Then we have pileated woodpeckers, cardinals, very interesting native butterflies. We have both non-native honeybees and native bumblebees. We have non-native worms and possum. So possum is another interesting species in that possums have been moving from the south to the north because of climate change. And so all of a sudden we had a northern species coming south, a southern species coming north, and they're overlapping in this little urban park, which has a weird conglomeration of plants. And so I realized that I had inadvertently been working for years in a novel ecosystem. And through iNaturalist, I was able to put all the pieces together to share the story of how a novel and unusual ecosystem can still contribute to the ecology of a local area and make it really quite successful. So um, I think that is it. So I will move forward with my happy slide and my iNat at if an iNaturalist app. So if you want to point your phone at that, you can join today, and I will happily field questions.
Amazing. I just love the way you explain how people and place and data are connected. So whether you are in Rochester, Minnesota, or across the Great Lakes Basin, thanks for chiming in, for learning from Angela. And the good news is also that we are recording this webinar, so those who did miss it can still learn from you, Angela, over the next weeks and months. Please also keep in mind that our project, the Sea Grant Great Lakes BioBlitz, officially starts on Earth Day, April 22nd. So yes, we'll get to ask Angela some questions. Yes, we will explain more on how to really dive into to get connected with iNat, but we also are interested in just a quick poll. So if you wouldn't mind, if you are joining us live, we'll put that poll up for you. We want to know where people are coming from. Thanks for taking a minute or two to do that. Also, after you've completed the poll, first question being the state where you're located. Second question, just typing in your city. And third question, your zip code. That helps us get a little more data for future webinar use. But also, if you have questions for Angela, please feel free to put those in the Q&A section. And on that note, thanks again, Angela, for sharing your stories and expertise. And I'm also wondering, well, I notice your playful attitude too, and I really appreciate that. Angela, what is it about iNaturalist data collection that is just fun for you and the groups you work with?
Yeah, it's a great question. So, I mean, professionally, I use it to gather data and empower community to help us answer these super tricky questions. And so it's awesome at that. But personally, I just use it to learn all the time. And so I didn't say this, and that was a miss, but iNaturalist works anywhere in the world on anything that's ever lived. And so I think about plants, but it can work scat, it can do insects, it can do worms, it can do cones and seeds. And so you can follow my vacations with my iNat posts. So if I go to a new area and I'm hiking or I'm camping or I'm doing anything, and I go out and I see something, oh, I don't know that one. Or I do a lot of invasive species. My husband is from India. We travel to Asia a fair bit, and I will see invasive species in their native range. And I'll be like, wow, I feel like I know that, but it looks a little different. And then I'll be iNatting again, right? And so iNat, I really enjoy it as a way to learn in live time about the world around me. And so, and I know that that can also help. I have no idea if there are scientists in those different countries or places that are actively looking at these reports, but now it's been documented. That documentation has been saved. And if anyone is curious about this, they can find it today or later.
Angela, I agree with you about the traveling. I also have had the joy of traveling, and it's really fun because you go to these ecosystems, you have no idea what's going on. So, you said in your talk about good photos, and I've been BioBlitzing on the Great Lakes BioBlitz, and I've taken some really bad photos. So, I would love to hear from you what makes up a good photo so we can get to research grade.
Yes, I'm glad you asked. So there are a couple of things that matter from my perspective. So I will move forward and show a very short video about 1 minute about good photos from iNaturalist. But before I do that, I think a good report in iNaturalist has 3 to 5 photos for every report. And so if it's a plant, we need the whole plant. We need something for scale because the size of the plant or the size of the leaf can be the difference between one species and a different species. So whether the scale is your foot, whether it's your finger, whether it's your kid, we need something for scale. And then we want different parts. So the whole plant or the whole species, and in plants, we commonly want, is it alternates or opposite leaf attachment? We want pictures of the whole leaf. If there are any flowers or seeds, those can be very definitive. If you're taking pictures of like an insect, and I realize, bees fly away, this can be tricky, but you want to get the front and the back and the sides and maybe multiple pictures if you can. And what happens is, so I think, uses the first picture. So move the best picture and the most unique identifying picture first in your AI queue, and that'll help iNaturalist get the verification correct. But then the humans that do the next level of verification, we look through all the pictures and we make sure that they are in alignment. So one, I think you should have a minimum of 3 to 5 pictures. And then the other thing that I alluded to but didn't say was the cultive or captive feature. So that's a choice in the or the iNaturalist observation. And as you can maybe imagine, in the invasive plant world, a tree, let's say a Japanese tree lilac, might be planted in a yard or on a boulevard. And in those places, it's a beloved part of that neighborhood. But that same species, if it escapes into the woodlot, like at Plummer House, then it might actually be considered invasive. So if I find a Japanese tree lilac or an amber cork tree on a boulevard, I would indicate that by saying cultivated. If I find that exact same species escaped into nature, I do not indicate that it's cultivated. And that can help folks like me understand whether it is potentially becoming troublesome. And so that feature is really important. But you asked about photos. So I'm going to advance my slide and show this really short. There's no audio, so you're just going to see the series and there's some words on the slides. So it's about 1 minute about how to take great identification photos. Awesome. Happy iNatting, everyone.
Thanks, Angela. Leah in our from one of the participants has a really good question, and especially getting to this idea of the research grade, because I know how important that is. She asks, when I look at comparing pictures on iNaturalist, are those only research grade species, or is it possible that they could be incorrectly labeled? So maybe like a little overview of the how they're ID'd on AI and then what happens next. That might be helpful.
Yeah, so I'll be honest, I think I understand the question. I'm not 100% positive of the answer, but I'm pretty confident. So how this works is when you go into iNaturalist, and I can just do that if you want me to. Like, I'm in iNaturalist all the time. And then we can really see what she's talking about. So here I am, and we're going to go Virginia bluebells. And I spelled it all wrong because, of course, I'm typing in live time, so that is always troublesome. So let's blue. There we go. So here you can see these, and there are many of these. So they have just become to emerge in my neck of the woods, and these are not yet to research grade, right? So this one is, however. And so if I go in, and what we're going to see here is that that it has. So likely the AI said, yes, it's a Virginia bluebell, and then two people agreed with it, and so now it gets this flag as as research grade. Now, if I want to go back and look here, I see all of these images, and these are the compare. So if you go in there, and it's going to show you these comparative images, and and I am pretty certain that iNaturalist only pulls research grade for these. And so when you're looking at those comparisons, then you really, I would be shocked if you're not looking at research grade, because iNat is pretty good about upscaling those research grades and using those. Now, that isn't to say that mistakes can't slip through, right? So, for example, when we're looking at oak species, the difference between a white oak and a swamp white oak is quite specific. And so we had to have special oak experts come in and do their best to get to that really specific species characterization, and try to only get those up to research grade. And so what that meant was there were some images we simply did not have enough good pictures, and they just linger out there because we can't get them up to research grade. So to answer the question, I do believe when you look and you're comparing, those should all be research grade. My experience is they do tend to be very accurate, but there can be inaccuracies even within the research grade category.
Okay, that made me really helpful. And Leah says, thank you. That answered her question. And I've had observations that then create a lot of conversation
Mhm.
from different experts, which is really, I think, what you were alluding to when you had that NASA observation. And so I've been doing this a while, but do you have one or oh, I forgot to say, my name is Ann Moser. Thank you thank you for Heidi for reminding me. I work for Wisconsin Sea Grant as education coordinator. But for those that are new to iNaturalist and making these observations, do you have one or two piece of advice you want to share that people shouldn't miss?
Well, first, just do it, right? Like download the app and go out there and do it. And I suspect you'll enjoy it. So we get a lot of converts through all kinds of things through iNaturalist. Don't be afraid. The app is easy to use. They're actually upgrading the app. So if you go to this QR code, this is a smart QR code. So in theory, it knows exactly what phone you're on, and it's going to give you the newest app. I'm an Android user. My understanding is they've actually updated the Apple platform, and it looks a little different than in some of the like older tutorial videos, but it is more user friendly. They're trying to really make it very user friendly. For Android, that has not launched yet, so that is still in the works. So my first bit of advice is download it and go do it. Um, I there is also a platform called Seek. Seek is for children. It's a wonderful platform for children, but it's really targeted for children in that the geolocation for internet privacy is turned off. And that is makes perfect sense for youth that we want to be careful about them. But those reports are less useful from a scientific perspective. If I don't know where it is, it doesn't have the same value. And so for grown-ups, I strongly, strongly recommend iNaturalist and not Seek. But no, if you want to iNat as a family activity, which can be quite fun, you can, your kids can have the Seek and the grown-ups can have the iNat, and you can all see who can find the most species. You can do things like that. But first, try it. And second, it takes less than a minute. And so, I tell people when I do invasive species reporting, a lot of people, they just are a little embarrassed. They don't know what it is. So iNat it. And if it's an invasive species, and then you report it to EdMaps, which is our invasive species reporting platform in Minnesota, you've done a great service by finding an early detection species quickly and reporting it. That's exactly what we want. So you got the ID wrong. You've just learned about a new native, right? Like that's the worst thing that happens. And that's a delightful outcome. So give it a whirl and enjoy learning while you're out there.
Such powerful advice. Yes, try it. And it takes less than a minute. So you get to learn something new by exploring. I love that. Thanks so much, Angela. I appreciate your help with this. we're just going to run through next just a few basic slides for those who really haven't tried a BioBlitz before or iNaturalist before. So know that in the next minute or two, we're just going to share some basics with you. So, and to recap, of course, like why iNaturalist? It's participatory science. Yes, this is a database to document any organism, understory, trees, insects, other critters that you see. So we can use this participatory science database across the Great Lakes Basin. It was originally developed by the California Academy of Sciences and it's supported by the National Geographic. It's now its own nonprofit. Also, please keep in mind when we're thinking about iNaturalist that it does allow for easy data collection, for species cataloging, analysis of data. So that collection experience is for you, but you are doing participatory science, so that data is easily shared. When you take a look at this collection of pictures, the photos you take for iNaturalist can be both fun and informative. So thank you, Angela, for sharing those tips and tricks about taking good photos and making sure we have enough photos for the photography with iNaturalist. That's just important to keep in mind. You don't know what you're going to see, but there is a way to identify something if you don't know what it is too. So it's a fun way to explore. And photography is a hobby. Chime in, right? So how do we participate in the BioBlitz? So first, what you're going to want to do is go to the iNaturalist on your computer or download the free app onto your mobile device. Then you create an account. And from your phone or computer, search for, and I put it in bold here right in the middle of the screen, so you know to search for 2026 Sea Grant Great Lakes BioBlitz. That is the project that we want you to search for. And then you'll scroll down and click join. Now, to what Angela just said, how they've updated the app. I have an iPhone and I just did this just yesterday and checked in. Last year, I wasn't able to search for a project on the app, but this year it is. So the updates are really helpful. You can use your desktop version or the app version to search for that project, 2026 Sea Grant Great Lakes BioBlitz. And once you've joined the project, you just start taking pictures with using the app. You open it up and photos and things pop up for you. It's a wonderful experience. So I encourage you to give it a try. Let's take a look at the next slide here. Just so you know what it might look like on a desktop computer, you would find that project in the app or online. So there's just a little search bar, and I put in 2026 Sea Grant Great Lakes BioBlitz, and then step 2, you'll see the page for the Great Lakes BioBlitz once you join, and then step 3, get outside, give it a try. Some people choose to search for something every single day. Others, we heard, well, while you're on a trip or vacation, all are good, but do please give it a try. And remember that our Great Lakes BioBlitz officially opens, the project opens on Earth Day, April 22nd, and runs for 4 weeks. Let's take a look at just the last slide here as a recap about our project. It's open to all ages across the Great Lakes Basin, runs for 4 weeks. That BioBlitz is a way for us to collect the data, identifying as many wild living things as possible in a specific area over a short period of time. And our area is the Great Lakes Basin, so we need your help. And then to submit, we're using iNaturalist. And there's more information on the Center for Great Lakes Literacy website, so you can go check it out there as well. Well, in conclusion here, it could be your backyard. It could be your school, the lakeshore, or your local park. Just try it. Try iNaturalist, join the 2026 Sea Grant Great Lakes BioBlitz project. This is participatory science, very user-friendly. Happy iNetting. And before we go, let's just give an extra special thank you to Angela Gupta for sharing her expertise, the stories, and also those tips and tricks. Thanks so much, Angela. I look forward to seeing you and your observations across the Great Lakes Basin on iNaturalist. So thanks, everyone. Also, thanks to all participants and our project partners, and also you, Fred, behind the scenes working the webinar. So thanks, everyone, for joining us.
Yeah, thanks everyone